Assalamualaikum warahmatullahi wabarakatuh. Hi everybody, we met again. I hope all of you in a good health. So for today's session, we begin Umul Kitab Al-Fatihah. Okay, so this week would be your last case study, final case study, which is case study number 6. That would be focused on coach postulate. So I have a video to share with you. So I hope that this video will help you to have better understanding in the coach postulate procedure. So after you finish watching the video, if you have any inquiry, please let me know. Thank you.
How do we know the causes of disease? The answer to this question started some 130 years ago with a German scientist named Robert Koch. Now, his specific interest was with an animal disease, anthrax of sheep. And in his observations, he had observed in diseased sheep the presence of a bacterium, whereas in healthy sheep there was no bacteria. But how did he prove that this organism caused this disease? As we will go through in this series of videos, he developed a series of steps that will allow you to conclude that a specific disease is caused by a specific pathogen. Now, even though he worked only with sheep, we in plant pathology now find that this series of steps that he developed is a fundamental concept used in determining the cause of all plant diseases. You will be able, at the end of this, to not only describe the steps of Koch's postulates, but also logically describe the reason you follow this series of steps with each of the plant pathogen types that we will work with in this class. So what are those 4 steps of Koch's postulates that we use in plant pathology? First is to establish a constant association of a specific pathogen with the occurrence of a disease. Secondly, is to isolate that pathogen into pure culture free of all other organisms. Third is to then take that isolated pathogen, inoculate a plant of the same species or variety, and reproduce the symptoms that you originally observed. And then finally, take that diseased plant, re-isolate the pathogen, and compare it to the original pathogen, proving that it was the same and thus the cause of that disease. Watch the following videos to learn the detailed steps involved in completing Koch's postulates. Step 1 of Koch's postulates is to establish a constant association between the presence of a pathogen and the occurrence of a disease. Now, what this means is that we need to examine multiple specimens and consistently find the presence of an organism. This does not prove that that organism causes the disease, simply it establishes that association. In this example, we will be using a disease of apple known as bitter rot to work through the detailed steps of Koch's postulates.
When deciding what specimen to collect, make sure to look at the range of disease symptoms present and select a specimen that has both diseased and healthy tissue present. Be sure to avoid collecting completely dead samples, as it is difficult to isolate pathogens from dead tissue. Be sure to take detailed notes, especially if this is a disease or symptoms that you have not encountered before. And if you have a digital camera, make sure to take good pictures.
So now that you've documented the symptoms, taken careful notes and images, you're ready to proceed to step 2 of Koch's postulates. After establishing that constant association between the presence of a pathogen and a disease, the second step is to isolate that organism into pure culture, which means we free it from the presence of all other microorganisms. This is an example of a pure culture of a plant pathogen, and it is the objective of step 2 of Koch's postulates. We will now take you through the series of steps required to obtain this pure culture.
In order to complete step 2 of Koch's postulates, we'll need our symptomatic apple, 95% ethyl alcohol, matches and an alcohol burner, 10% bleach solution, sterile water, Petri plates containing a growth medium, parafilm strips to wrap the plates, forceps, scalpel, Sharpie, and paper towels. The purpose of sterilization is to minimize contamination from other microorganisms that might be present. We'll start by sterilizing the surface of the apple by taking a paper towel and dipping it in to 95% alcohol and gently wiping the surface from which we're going to isolate. Next, we're going to sterilize the scalpel that we're going to use to cut out diseased tissue. Dip it into the 95% alcohol, let the excess drip off, then hold it over the flame. The rapid burning of the alcohol helps to kill microorganisms that may be on the surface of the scalpel. It's important to keep the scalpel, now that it's sterilized, from touching other non-sterile surfaces. Using the sterile scalpel, we're now ready to collect symptomatic tissue from the apple. Be sure to collect tissue from an area that contains both diseased and healthy tissue, as the pathogen is most active at this advancing edge of the lesion. Remove several small pieces from the edge of the lesion. Once we have several pieces of tissue, we need to surface sterilize them. This step should kill all organisms except for the pathogen that is growing inside the tissue. The first thing we'll need to do is sterilize the forceps. Dip them into the alcohol, drip off the excess, and then hold it into the flame. Use the sterilized forceps to place the pieces of tissue into 10% bleach solution. They will sit in the bleach solution for 30 seconds. Using the forceps, remove the pieces from the bleach, rinse them in sterile water, and then place them onto sterile growth medium. Use parafilm to wrap the plates so that the medium does not dry out during incubation. It's a good idea to label your plates with the date and sample ID. After a week, this is the growth that we see on our plates. You'll notice that all 3 of our isolations have grown out and are similar in appearance. The next step is to transfer the organism to a new plate of medium so that we ensure a pure culture. For this next step, we will need a sterile scalpel and a fresh plate of growth medium. Sterilize the scalpel as before. We'll need to isolate from the edge of the colony, since that is the area where the pathogen is most actively growing and where the least amount of contamination is likely to be. With the scalpel, make a cut at the edge of the colony and remove a piece that includes both the colony and clean agar. Then transfer that plug over to a new plate. Then parafilm and incubate as before.
Okay, so now that we've established that association, we have our organism growing in pure culture, now we need to put it back into the same plant. And this is step 3 of Koch's postulates, where we re-inoculate the same plant, species, or variety with the organism that we now have in pure culture. The goal is to reproduce the symptoms of that disease on our newly inoculated plant. Now that we have a pure culture of the organism from the diseased apple, we need to complete step 3 of Koch's postulates, where we determine if this organism is capable of reproducing the symptoms of the disease in a healthy apple.
For this step, we'll need a scalpel, alcohol matches and an alcohol lamp, an apple of the same variety that we isolated from, paper towels, the pure culture that we've isolated, parafilm, and a digital camera and notebook. You'll need to document the appearance of your pure culture. To do this, you can either describe it or take a photo that you can insert in a lab notebook. We are going to inoculate the apple by making several small wounds and then putting plugs of the hyphae into those wounds. By wounding the apple, we are creating favorable locations for infection to take place. Begin by surface sterilizing the apple with alcohol. Then sterilize the scalpel. Now we'll make several small wounds. Make sure you mark which wound is going to be the control and which ones are going to be inoculated with the pathogen. The first thing we'll do is flame sterilize the scalpel again. Now in one of the wounds, we are going to put clean agar that does not have any organism growing in it. This will serve as our control to ensure that any effects are caused by the organism and not by any of the procedures that we are performing. So take a small piece of agar away from the colony. And then carefully place it in the wound. Then reflame the scalpel. And this time, we're going to take plugs from the colony and place them into the other wounds. Flame the scalpel again for the next plug. We will now put parafilm over the inoculation sites to ensure a favorable environment for infection. We will now incubate the apple and observe daily for symptom development.
Now that you've reproduced the symptoms of the disease, it is time to complete step 4 of Koch's postulates. In step 4, we re-isolate from that symptomatic tissue and compare that organism to the organism we obtained in step 2. Now, if those organisms are the same, you have now completed Koch's postulates. You have isolated, inoculated, and then re-isolated the organism from the disease. Now, by completing these steps, we have proven that this organism is the cause of this disease, and you have completed Koch's postulates for this pathogen.
After reproducing the symptoms of the disease on the inoculated apple, the next step is to isolate from the diseased tissue and determine if the organism that grows out is the same as the original isolation. We've isolated from the inoculated apple, and when we compare it to the original isolation, we can see that the organisms are the same.
Now that you've confirmed that the re-isolated organism is the same as the original, you have proven that this is the cause of the disease. And congratulations, you have completed Koch's postulates.
So make sure before you write the report, you watch the video given or the video upload in iLearn, and then you have to understand the Koch's postulate steps. Then you require to simulate the similar step or similar procedure to diagnose anthracnose disease of chili. So anthracnose disease of chili usually caused by Colitotrichum capsicum. So you may use the figure from the internet to fulfill the 4th step of Koch's postulate, and then prepare the report with the following format. So all the format is similar to the previous report, except body of report. So for results, for the first step, disease associated with the causal agent, you have to explain about how the possible pathogen could be associated with the disease, and then you have to put the related figures that you obtain from the internet. So the figures should be on chili that have anthracnose symptom. And then step number 2 is solution of causal agent. By right, you should know what are the causal agent of anthracnose. So just for simulation, we figure out that the possible causal agent of anthracnose is Colitotrichum, and then you can find what are the morphological characteristics of Colitotrichum on the media. Alright, and then you can use the figures from the internet and put on the the report. And then inoculation. So in this step, inoculation, you will use the pure culture, or in effect use the pure culture, or in fact the pure culture to the healthy plant or healthy fruits. So what will happen to the healthy fruit when we inoculate it with the pathogen, possible causal agent? Okay, so you have to show what is the symptom. Of course, you can use figures from the internet. And then the last one, re-isolation. So from the symptom that you obtain in the step number 3, so you re-isolate and then you compare the isolated culture with the pure culture. So just answering for a question in discussion. Okay, I think that's all for instruction for report writing. Okay, that's all for today. Thank you so much. Assalamualaikum warahmatullahi wabarakatuh.